Introducing symbiosis. It will not be a textbook definition, but rather my own appraisal of how different creatures live together in pairs, trios, quartets, right up to the community level. Here we see a plant above ground, but it descends into the soil where it is plugged into a fungal network, which in its turn is attached to tree roots. A triple symbiosis, most of it beneath our soon-to-be illuminated feet. Whether we look at a single plant or whole communities of plants, for instance woodland, what do we see? Maybe we focus on just one plant or look around to observe all plants, one species or many species at once. What could we see if we used our brain to synthesize in the imagination, in detail, a pieced together, multidimensional image of an individual plant, then plant assemblages, and then whole interactive communities above ground and below ground, consisting of plants, fungi, bacteria, animals, and so on? There's a lot we must take in if we want to understand our natural surroundings. When thinking this out, we should cast our minds above ground and below ground while also considering interactions going on between them. If possible, we must make good use of all the traditional and modern tools we have to hand: fieldwork, lab work, microscopy, DNA analysis, statistics, thought, and ultimately enlightenment. Given time and information, that sort of profound understanding can be achieved, but never completely. At our present point in the constantly improving enlightenment of Homo sapiens, we know an awful lot, much more than every generation of our ancestors, but whether we will ever know everything is doubtful. That is not a deficiency to give us fear or regret, but a welcome challenge automatically provided by our innate human curiosity, which, because of the natural world's intricate complexity, is an endless quest we can pursue and appreciate and enjoy. There's so much we can gain from learning about what's going on beyond our ordinary senses, looking elsewhere for information that will enable us to understand and value our surroundings. One major feature of ecological communities is symbiosis. All but the most primitive of organisms are symbioses, all manner of species collaborations, any of which we might consider to be a community in itself. Humans, for instance, are the body we see, plus our gut community of bacteria and single-celled creatures, the protists, and the mitochondria in each cell that once were bacteria that took up residence in other bacteria long before multicellularity had evolved. Then there's all that lives on our skin, in our various orifices, and so on. Symbiosis is simply everywhere. Here, in fast-flowing rivers where salmon breed. Freshwater pearl mussels depend entirely on salmon reproduction to facilitate their own. No salmon, no pearl mussels. What is symbiosis? Briefly, symbiosis consists of two or more organisms living together with at least one of them gaining some benefit. Thanks to symbiosis, some fungi can grow on the most unlikely substrates if they collaborate with another organism, in this case a cyanobacterium, which can manufacture and share carbohydrate which the fungus, in this form known as a lichen, is unable to obtain unaided. The fungus provides somewhere for its partner to live, as well as being able to gather nutrients to share, supplies of which on a road sign are likely to be scarce, but nonetheless just sufficient. As demonstrated on the road sign, lichens are great ecological pioneers, here exploiting an inhospitable rock surface on which plants may grow later, just as the moss is doing. A similar process occurs on tree bark. Here the pioneer lichens are wafer thin, and here taken over by a foliose lichen, moss and liverwort community. In lichens the benefits of symbiosis tend to be mutualistic. We, observers of nature, have applied the term parasitism to relationships that could be, if judged less harshly, fairly be termed symbiosis, such as this honey fungus and its birch tree victim, which it will kill and eat. But no matter how cruel, parasites do not eliminate populations of their victims, many of which survive unaffected, and the communities in which they live benefit from the ecological controls that parasitism provides. Others may disagree, but I count parasitism as symbiosis, beneficial at the community and habitat level. Here, to its benefit, the parasitic barnacle Saculina has prevented this female crab from molting, enabling it to feed on its unfortunate host until it's done with her, while the opportunistic barnacles, because they won't be sloughed off during a routine molt, have the opportunity to remain on the crab's carapace and multiply. Symbiosis consists of at least 5 escalating functions. 1. Interaction, living together. 2. Interdependence, partnership. 3. Iteration, repetition. 4. Intricacy, multiplicity and interweaving, and then the marvellous emergence of the complex communities we call ecosystems. In this example, a lion's mane jellyfish, travelling upside down, provides a protective environment for a shoal of little fishes, safe from predators among its stinging tentacles. The relationship may seem simple, but it is a mere component in the infinite complexity of the marine environment. Interaction, living together. Here is a lichen, but not as we know it. These little mushrooms belong to a fungus that lives in sphagnum tussocks, but is sharing its lifestyle with a single-celled green alga growing unseen on and in the moss. Here's another very familiar example of symbiotic interaction. Interdependence. Once a symbiotic relationship has become established, there is usually interdependence built into the system, with benefit accrued by both partners, but that's why symbioses evolved. Here, in a modern botanical setting, bluebell roots are packed with the fungus Scutellespora dipurpurescens, which provides phosphate scavenged from soil in exchange for carbohydrate the plant manufactures from photosynthesis. Neither can perform the other's nutritional function. Neither can thrive without the other. Interdependence. This is a form of mutualism. I've now mentioned mycorrhiza, and since it's going to be a main topic later in this presentation, I'd better explain what it is here. The term is a combination of 2 Greek words, mykas and rhiza, so the definition of the symbiosis is in the name, fungus root. Mycorrhiza is the means by which green plants and specialized soil fungi contribute to the nutrition of each. Plant roots are invaded by distinctly beneficial fungi to form the symbiosis. Thus, mycorrhizas, not roots alone, are the chief organs of nutrient uptake by land plants. The sun shines on the plant, which manufactures carbohydrate the fungus is unable to synthesize for itself, contributing about 20% to the mycorrhiza, thus feeding the fungus. The fungal partner is able to access low concentrations of soil phosphate, which the plant roots can't, transporting it into the roots and thus into the mycorrhiza, feeding the plant. With that important definition completed, albeit far too concisely, we will return to the emergence of complexity. Iteration. Repeat, 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 etc. A good idea will evolve by repetition as well as by change. In the spring, ants feed on the sugary secretions of nectaries, green pads by the joints on bracken stalks. Early in their evolution, ferns and ants began to cooperate, and now, by iteration of the idea, many ants and plants do, while the relationship has diversified. Intricacy. Symbiosis is interwoven with others in intriguing but quite complicated combinations. The Indian pipe is a flowering plant with no chlorophyll, so it cannot produce its own carbohydrate as most other plants do. It forms a 3-way partnership below ground with forest trees and their mycorrhizal fungi. That we call mycoheterotrophy. Here that is explained. This photo montage shows what we might see in an ideal field situation, with the tree on the right, the Indian pipe centre, and the mushroom of the soil fungus left. In reality, they will all be present, but aren't necessarily seen together like this. Let's dig down into the soil. The Indian pipe has no more than a cluster of stumpy roots attached to the mycelium of a soil fungus, which in its turn is attached to feeder roots of nearby trees. The fungus and the tree are associated as a mycorrhiza, one function of which is to supply the fungus with carbohydrate manufactured by the tree by photosynthesis. The fungus can't do this, and neither can the achlorophyllus Indian pipes, so both receive a supply from the tree. Wikipedia and other online advisors may describe this relationship as parasitism, but no, emphatically no. It is a co-evolved cooperation from which all three partners benefit and none is harmed. The non-judgmental term mycoheterotrophy, meaning feeding via a fungus, is much more appropriate. Here is just one more escalation in the intricacy of symbiotic relationships, mixotrophy. Like the pale achlorophyllus plants, the monotropa and others, the broad-leaved helleborine is plugged into a mycorrhiza of adjacent trees, here probably downy birch. But it's green and fully capable of producing its own carbohydrate by photosynthesis, but it grows in the deep shade of woodland. Its reliable, tree-donated carbohydrate supply enables it to grow where light, and therefore its own photosynthesis, are limited. Yet another complication occurs when a mutation creates an albino helleborine. Now it is in the same predicament as the Indian pipes and is able to cope in the same way. It has no chlorophyll, so it should starve, but because it has a reliable carbohydrate supply donated by trees via their mycorrhiza, it can continue to grow and is not uncommon. This perhaps demonstrates how achlorophyllus plants might have evolved. Here are two more British orchids that live in a three-way symbiosis with trees and mycorrhizal fungi. One has chlorophyll and the other does not. By emergence, we are referring to the gradual development of complexity from initial states of simplicity. It is a fascinating, if complicated, process detected not only in nature but also in many other circumstances. Emergence is the focus of a new scientific discipline, recognised properly and studied only relatively recently at the Santa Fe Institute. If we count a one-to-one symbiosis as simplicity, say of this mature oak and a root-inhabiting fungus, then complexity emerges as the diversity of fungal species in the roots increases during the lifetime of the tree. Adjacent plants join the local community, many with their own different fungal associates, at least 11 with bluebells alone. And the next generation of trees, seen here surrounding the mother oak, all developing their own communities of root fungi, and so much more going on. That was a long and complicated sentence, unashamedly presented to describe a long and extremely complex ecological process. There is an endless supply of symbioses waiting to be discovered and described. Here's a possible candidate: otters and the juvenile phase of palmate newts, called efts. Otters routinely emerge from the sea to wash their fur in freshwater pools above high water mark. They give away their presence by depositing their spraint in habitual latrine areas, often next to wash pools, which become tussocky and bright green. The water of wash pools can become quite, shall we say, organic, an ideal habitat for microbes, suitable food for efts. So there might be a research project in the question: is there a symbiotic relationship between otters and newts? Having now established some of the background biology of symbiosis, let's turn our attention to the bluebells and the symbiosis that we know supports them, as well as most other plant species, at every temporal and ecological level. Mycorrhiza. In its various forms, it's all over this picture and in any other landscape, a fundamental part of the scene, but we can't see it. Below ground, in soil and roots, and microscopic. The plant-fungus symbiosis mycorrhiza, in its different forms, has been on Earth for 400 million years, is the normal lifestyle of most plant species, and occurs on all continents. It is, therefore, absolutely fundamental in our understanding of the lives of plants and their special fungi, and in consequence the complexities of terrestrial ecology. When engaged in landscape destruction or its restoration, we really need to bear this in mind, wouldn't you think? Next, we'll consider the four main types of mycorrhizas underlying this scene and their place in the landscape. First, just the big oak, which is ectomycorrhizal, as would be several other tree species if they were in the picture. The ectomycorrhizal system is the only one of the four that we can detect by above-ground manifestations, mushrooms. The fungus root association is invisible below ground, but can be seen after some careful digging, centre. The proximity of certain mushrooms sometimes shows, as here, which partnerships have formed. Scenes like these, admittedly cherry-picked, provide us with some perception of the relationship between the above and below ground realms, and with the application of a microscope, experiments and imagination to piece together the fragments of knowledge that we do have, we can begin to understand the entire system. Next, the meadow consisting of numerous species, many of them arbuscular mycorrhizal. That includes the bluebells. Orchids present will have their own special mycorrhiza, while some species will have developed strategies for doing away with mycorrhizality altogether. Those exceptions are fascinating, and I'll discuss them in another video. A good number of trees in the region are also arbuscular mycorrhizal, and in this picture that includes hawthorn. Gorse, which paints the hillsides bright yellow in spring, is also arbuscular mycorrhizal. The open hillsides, where soils consist mainly of peat, are very different. Phosphorus is not the main limiting nutrient, whereas nitrogen is, so many plants growing there require ericoid mycorrhizal fungi, which can mineralize the organic peat, releasing inorganic nitrogen compounds that the heathers, bilberry and other ericaceous plants require. Also, there will be orchids with their special needs, and herbaceous plants and grasses that associate with arbuscular mycorrhizal fungi with varying commitment. With sufficient understanding, we can read a landscape. Given knowledge of the underlying geology and the soils created, and assessing plant communities even from an informed glance at a distance, we can determine which mycorrhizal communities might be present, and then why there are different habitats. An exercise I conducted in real life when objecting to the landowner's desire to plant trees all over this mostly treeless landscape. Trees don't grow where trees can't grow, if among other controlling conditions, the required mycorrhizal fungi are, predictably, likely to be unavailable. Now we'll turn our attention to the bluebell and its mycorrhiza. Normally we see only what is above ground, while unseen there's a lot going on below, so that's where we must look. But I'm afraid it's not going to be easy. A single bluebell plant has a bulb composed of compact white flesh and has at its base a dome-shaped disc of a stem upon which roots, two colourless leaf sheaths, green leaves and scapes, that's the flower stalks, are all borne. This old illustration of a single naked bluebell is remarkably accurate, except the root system, which is after all fundamental to this study, is shown unnaturally with all the roots short and dangling. In real life, the roots can be much longer and spread, many of them horizontally, with some diving straight down and others reaching up into the nutritious upper soil. In order for the French artist to paint this completely exposed plant, it would have been necessary to dig it up and wash away any soil adhering to the remains of the roots, which being brittle will have broken into hundreds of fragments left behind in the field. The roots remaining still attached to the bulb, being wet, will have tended to droop, adhering in a bunch. The artist has rearranged them neatly and tried to reconstruct the root system, but was unable to represent the root's true spread and orientations. The artist has also shortened the below-ground stem. Mature bulbs are positioned deep in the soil in the mineral horizon, where phosphate is in poor supply, hence the utility of mycorrhiza. Thomas Woodhead, in his day a renowned Huddersfield botanist, is one of the few people to have investigated the biology of bluebell. His drawing of a soil section shows the mature bulbs well below other woodland plants, bracken and soft brome. Bluebell roots inhabit a soil horizon in which phosphate is very scarce. Compared with, say, a grass, its roots are few, short and unbranched, not made for reaching out through the soil to gather a rare nutrient. From the earliest days of life on land, plants have always engaged with fungi, forming symbioses which overcome this nutritional limitation, mycorrhiza. A wide-ranging fungus can better forage for phosphate than its plant partner, which in its turn will assist the fungus with the product of its photosynthesis, carbohydrate. Many modern plants are completely reliant upon this cooperation, which has been field-tested over several 100 million years. Most bluebell roots for most of the year are packed with mycorrhizal fungi of the order Glomeromycota, here represented by photomicrographs of a glomus and a Scutellespora species. At our Yorkshire field site, it was Scutellespora that dominated the mycorrhiza from root emergence in mid-August until leaf flushing in late winter. Experiments showed that it was pumping soil phosphate into the plants via their roots at a time of maximum growth. From New Year, other species began to take over, their individual functions difficult to determine. Here is a section of root well colonised via mycorrhizal fungus. What might we see if we were to slice it in two and then swivel it through 90 degrees to look at the cut end? I don't have a picture of that, but rather conveniently there are pictures of 400-million-year-old plant fossils showing a similar mycorrhiza and telling an important story. Here we're dealing with the most ancient of several mycorrhizal symbioses, arbuscular mycorrhiza, which we know from fossil evidence and DNA studies co-evolved with land plants some 400 million years ago. Here's a cross-section of a plant fossil from the Devonian period, proving that mycorrhiza evolved along with the very earliest of the land plants. The white ring shows where the fungus has occupied the rhizome tissue. The arrow is pointing to one of many ancient arbuscules, highly branched organelles inside root cells where nutrients are exchanged between the plant and its fungal partner. This is it. Compare the ancient arbuscule with its modern descendant, here of the fungus glomus, in a bluebell root. Is that not extraordinary? The fossils were first described by Kidston and Lang between 1917 and 1921, and a lot of further research has confirmed the presence of mycorrhizas, promoting the theory that green plants and fungi evolved the terrestrial lifestyle together. Imagine the excitement that ricocheted around the mycorrhiza labs of the world. I was there at the time, when a Canadian team led by Luc Simon reported that their DNA studies confirmed the ancient origin of mycorrhiza, the date being coincidental with that of the earliest known fossils 400 million years ago. Before going any further, I think it's important to recognise that being mycorrhizal, the bluebell is anything but unique. The majority of plants on the planet are involved in one or another of the several types of mycorrhiza, while the few that are non-mycorrhizal have evolved mechanisms that have enabled them to opt out. However, we're here to look at just the bluebells and their mycorrhizas. Why bluebells? We must look at the components of nature before we try to understand how it works as a whole, and bluebell is the plant about which I know the most, having spent 12 years studying its ecology, above ground and below ground at the University of York. Unlike the example trees with mushrooms that we saw, there is nothing to see of the symbiosis that sustains Britain's favourite wildflower. But if you don't look, you don't find. First, you must dissect the subject of your inquiry. The senses and the mind must combine all the facts and intricacies of the unseeable, piecing together as many of the parts of an ecosystem that modern science can find out, eventually constructing an understanding of the whole. With permission, of course, for a whole year I dug up clumps of bluebells and took them back to the lab, where I divided each plant into its constituent parts: roots, bulbs, leaves and flowering stalks. The parts were sampled and assayed for fresh weight, dry weight, phosphate content and soil phosphate, while a root sample was taken for staining to show up the fungi inside, under the microscope to be identified and assayed for colonisation intensity. That created a lot of data which showed changes in the allocation of biomass and phosphate inflow, which in turn correlated with the quality and intensity of mycorrhizal colonisation. Some desirable investigations are just impossible, such as the format of the fungus outside the plant roots, in the soil where the fungus exists as a network of fine microscopic fibres too small to see. If you dig to have a look, the network is shattered, entirely losing its structure. You finish up with a handful of soil with nothing to see of these special fungi. Maybe one day someone will invent a method that overcomes this shortcoming. It would be very interesting. We can, however, sample bluebell roots which, after processing, we can visualize, categorize and quantify. The roots need to be rendered transparent and the internal fungus stained so that it can be examined with a high-power microscope. The method can be hazardous. When nearing the end of a career in which I used stains of sometimes appalling toxicity, a German lab discovered a superior and completely safe alternative. Vierheilig et al. Had discovered the usefulness of pelican ink, not known in the UK. During a formal teaching course, one of the participants was a fountain pen user, his pen loaded with Parker Quink. He provided the stain we needed, and the result was spectacular, so immediately we converted to Quink as our routine mycorrhiza stain. Having been cleaned, cleared and stained, roots are laid out neatly on microscope slides. Here are some of the slides made from root samples gathered throughout a growing season, August until July, ready for microscopy. The intensity of blue, Quink of course, shows just how packed these roots are with fungus, all the year round. Examination showed that various species colonised in sequence, providing various ecological services. Seen close to, we can see that September roots, only a couple of weeks since they emerged anew from the bulb, were already well colonised by what turned out to be the fungus Scutellaspora diperperescens. However, it is very, very difficult to differentiate the 11 or more fungi that associate with bluebells, though not completely impossible with practice and the help of a DNA sequencing colleague. It's even more difficult to name them. They do make spores of a sort which are highly characteristic of each species, but those are always found apart from the fungus in soil samples, so almost impossible to relate to the fungi they belong to, unless by chance a spore is clinging to a piece of root. If we can't apply names to the fungi, we can describe and quantify recognisable differences. In the autumn, roots will look like this, absolutely packed with fungus. Probably one we can identify with confidence, that's Scutellaspora diperperescens. By way of contrast, though, here is an unusual bluebell root that has not been colonised, while here is a root fully colonised by a different species of mycorrhizal fungus. Can you tell? And here yet another species of mycorrhizal fungus, from its structure, with those globose vesicles, recognisable as a species of glomus, but I couldn't say which one. This specimen has unusually retained some attached fragments of the external soil phase of the fungus, and we can see where it has entered the root in two places. The globular structures are oil-filled vesicles, a nutrient storage system. External hyphae enter the root by two methods. Mostly they clamp onto the outer surface of the root, cementing themselves in place by a pad known as an apressorium. The invading hypha drills into the root, growing into the main part, the cortex, where it proliferates longitudinally between the cells along the root. Less frequently, the invading hypha clamps onto a single-celled root hair, drills into it and grows down into the root. When it reaches the cortex, it exits the cell interior and, as is happening in the more usual sort of entry on the left, proliferates along the root between the cells. Here we can see the internal hyphae travelling along the root with fuzzy items off to either side. These are the little organs that facilitate nutrient exchange between the fungus and its host bluebell, the arbuscules, literally little trees. Here is an arbuscule in close-up, less like a little tree than some. Here is where the fungus provides the bluebells with phosphate from August until New Year, when other fungi take over. This sort of arbuscule is a thick, twisted coil bearing tufts of fine elements. These arbuscules are quite different, much more tree-like, and belong to a different fungus, the glomus species we saw earlier with its vesicles. Other than pushing through the cell wall, the fungus does not enter the cell properly, instead pushing into the membrane which lines the inner surface of the cell wall, the plasma membrane. Thus, every finest element of the arbuscule has intimate contact with the cell membrane, facilitating nutrient transfer into the bluebell, notably of phosphate, and this process has been going on for hundreds of millions of years, since the dawn of life on land. How do we go about conducting a field research project? Ideas first. We make observations and start asking questions. We share our thoughts, enabling an important ecological question to be posed, for which a practicable research strategy may be devised. My project, begun in 1989, was to discover whether the properties of mycorrhiza, already demonstrated under convenient artificial conditions in the greenhouse, represented what actually happened in the wild. My research plant was bluebell, a beautiful inhabitant of beautiful woodland, while my colleagues who set out to research a similar question, for which importantly they got different but equally significant answers, were given a small winter-flowering grass which grows in cold, wet, windy places in East Anglia. We measure and record every parameter we can think of, creating a vast spread of ecological data that may or may not be used later. The important thing is not to miss information we might eventually need. In my work, I first took samples for detailed analysis, in the first exercise carefully lifting the bluebells aside, removing some roots and replacing the plants. I also sampled soil from around the roots for the extraction of spores. Both samplings provided plenty of data, but unfortunately in practice the two did not correlate. Other results provided a lot more valuable information. At the same time, individual field-collected bluebells were divided into 4 parts to ascertain phosphate, P, inflows, and allocation of P and carbohydrate, C, to the various bluebell tissues, plus root colonization by mycorrhizal fungi throughout the seasons of a single year to demonstrate, or not, an ecological function of mycorrhiza under field conditions. That experiment provided the first evidence of mycorrhizal behaviour in an undisturbed natural ecosystem by demonstrating the temporal coincidence of root colonization by mycorrhizal fungi and the inflow of phosphorus in adult bluebells. There are numerous ways to find out how the bluebell mycorrhiza works. Here, a field experiment in which a fungicide was used to see how bluebells in the field get along without mycorrhiza. The answer? Rather badly, confirming the conclusion already reached from the physiological study that bluebells and their fungi are dependent upon each other. Question: how do plants, including bluebells, fare when inoculated with different mycorrhizal fungi? Answer: some extremely well, others, in particular the bluebells, very poorly. The bluebell mycorrhiza works at its best in woodland, left alone undisturbed, to complete its life cycle in the same place every year while spreading unhurriedly. My research at York generated a small cascade of peer-reviewed publications for other scientists to read. Also, in the magazine British Wildlife, presented to a wide audience of naturalists, the scientific papers would be unlikely to reach. And then to a very different but interested audience in Resurgence magazine, self-described as at the heart of earth, art and spirit, followed by a translated version in an Italian magazine with a similar non-scientific audience. Locally in northwest Scotland, bluebell mycorrhiza found its way into the Skye and Lochaushe Environment Forum's website and in 2023 this YouTube channel. We must keep thinking about what we know about bluebells and other biological unknowns and what we don't know, alongside many other ecological puzzles, extending our thoughts from the individual through the local to the global scale. Our researchers warned that if humans severely disturb their favourite wildflower or its fungal partners, yes, lab experiments and field trials showed that bluebells do not like soil disturbance, both will decline until they disappear, leaving us wondering why it all happened. If we don't do it by habitat destruction, climate change will probably do it for us anyway. That's a conversation we really must have. Of course, we know that is precisely what's happening. Biodiversity is declining worldwide. How could that be? Probably in many ways just human ignorance and greed, but unless we look deeply into the difficult science, we will never see it from nature's perspective. If we want to understand nature, and we really need to, this branch of the biological sciences, with others, is essential, indeed fundamental, but please don't expect it to be easy. Nature has had 4.5 billion years in which to become complex, self-sustaining, and, for us, difficult to comprehend, particularly if we don't try to find out. If we seek to understand nature, we will realise that it's not our job to exploit or restore it, but to give it space to function as it used to before we came along. Nature can be remarkably resilient if allowed to carry on without our interference. Here is the site shown in the two previous pictures, seen from the hillside above. There is mature woodland left and right of the gap, which not long ago was just bog and bracken, but now is filling in with new woodland, as is the slope in the foreground left. Over the past 20 years I have had the privilege of watching this happen. Just 20 years. Similarly, just a few miles away, I've watched the change from impenetrable gorse to species-rich birch woodland. Every stage can be seen in this picture, from early invasion by birch and rowan, 1, to trees overtopping and suppressing the gorse, 2, to new woodland, 3. The change is spectacular, if you watch. In 2015, 2 of us braved the thorns for a species count within the apparent gorse monoculture by the Skye Bridge. The previous photo was taken from the hilltop on the right. We were astonished to have recorded more than 160 plant species ready to become woodland as the gorse aged and fell apart, letting in the light, eventually dying. Bluebells taught me a great deal about their lifestyle and then the lifestyles of other plants and fungi and the intricate ecological communities in which they live. However, my knowledge is but a snapshot, and my ignorance emerges as soon as people start asking me questions with the expectation of definitive answers. Sorry. Because my understanding of this hugely complex topic is limited, we soon get bogged down in the hypothetical and must all deal with uncomfortable uncertainties. But let's talk anyway, because that's the way to discover new questions that maybe can be answered, eventually.